All right, let's talk pharmacology. If there's one topic that you've got to nail for your exams, it's the cholinergic system. So in this explainer, we're going to skip the fluff and get right to the good stuff, the high-yield facts that you'll see over and over. Ready? Let's jump in.
So let's start with a bit of a pop quiz. Out of all the neurotransmitters we talk about, dopamine, serotonin, the whole crew, which one do you think was discovered first? Well, the answer is acetylcholine, or ACH for short. And honestly, its discovery is the story of how we even figured out that our nerves talk to each other using chemicals in the first place. It's that fundamental.
Okay, so here's our game plan. First, we'll look at how it was all discovered. Then we'll walk through the entire life cycle of acetylcholine. After that, we'll tackle the big one, nicotinic versus muscarinic receptors. Then we'll map out its pathways in the brain and body. And we'll wrap it all up with a super high-yield rundown of its organ effects, perfect for your exam prep.
All right, section 1, the groundbreaking discovery. Seriously, understanding this origin story is key because it sets the stage for literally everything else we're going to cover about the cholinergic system. You can basically thank these two guys, Dale and Lowy, for the entire field of neuropharmacology. Back in 1936, they won the Nobel Prize for proving that nerves don't just zap each other, they talk using chemicals. But their real breakthrough, the moment that changed everything, was realizing that acetylcholine wasn't a one-trick pony. It didn't just do one thing. It could have two completely different, even opposite effects. And the way Lowy figured this out was just brilliant. So he gives an animal some ACH, and its blood pressure plummets. That's a classic parasympathetic response we now call a muscarinic effect. But then, here's the genius move. He blocks those receptors with a drug called atropine. Then he gives a huge dose of ACH, and guess what? The heart rate actually goes up. This totally unmasked a second nicotinic effect, and was the smoking gun that proved there had to be two totally different types of receptors. Okay, so we know what acetylcholine does. Now let's get into the nitty-gritty of how the body handles it. We're going to follow its whole life story, from the moment it's born to how it does its job, and then how it gets cleaned up super fast. Thinking of it this way really helps you remember the key enzymes you'll be tested on. This image here is basically our map for the whole process. It shows you everything that happens in the cholinergic synapse. We're going to use it to follow a single molecule of acetylcholine on its journey, right from its creation on this side all the way to its effect on the other side. Let's walk through it step by step. So it all boils down to this four-step dance. Step 1, synthesis. A choline molecule gets pulled into the neuron, and this enzyme, CHAT, you gotta know that one, choline acetyltransferase, slaps it together with acetyl-CoA. Step 2, storage. It gets packed up into little bubbles or vesicles by a transporter called VACHT. Step 3, release. An action potential comes along, zap, and the vesicle dumps the ACH into the synapse. And finally, step 4, metabolism. Almost immediately, it gets chewed up by another key enzyme, acetylcholinesterase, or ACHE. The leftover choline then gets recycled right back into the neuron. Super efficient, right?
Now, here's a classic exam trap you need to avoid. There are actually two types of cholinesterases. You've got ACE, which we can think of as the true one. It's the specialists living right there in the synapse, ready to shut down the signal. But then there's this other guy, butyrylcholinesterase, or BCH. Think of it as the pseudocholinesterase. It's just kind of floating around in the blood plasma. And for your exams, the big thing to remember is that BCH is the one that chews up drugs like succinylcholine. Super important distinction.
All right, we've made our neurotransmitter. We've released it. Now what happens when it gets across the synapse? That brings us to section 3, nicotinic versus muscarinic receptors. Think of these as the two different kinds of locks that acetylcholine's key can fit into. This is one of the most fundamental concepts in all of pharmacology, so really lock this in. Nicotinic receptors are what we call ionotropic. That's just a fancy way of saying they are the ion channel. ACH binds, a gate swings open, sodium floods in, and bam! You get a super fast, direct electrical response. Muscarinic receptors, on the other hand, are metatropic. They're G-protein coupled. So when ACH binds, it doesn't open a channel directly. Instead, it rings a doorbell that kicks off a whole chain reaction inside the cell. It's a slower, more indirect, and more, modulatory effect. So remember, fast and direct versus slow and modulatory. Let's zoom in on the nicotinic receptor for a sec. You can see it's this big protein complex made of 5 different parts, or subunits, all arranged in a circle to form a little tunnel down the middle. But here's the absolute key takeaway for your exam. It takes 2 molecules of acetylcholine to open this gate. They have to bind at the same time, one on each of the 2 alpha subunits, for that channel to pop open.
So where are these things? Well, for your exams, you gotta know these key spots. The neuromuscular junction, that's how your muscles contract. You'll find them in all the autonomic ganglia, both sympathetic and parasympathetic. They're in the adrenomedulla, and of course, all over the brain. The effect is always the same. Sodium rushes in, causing an excitatory depolarization. And here's a clinical pearl for you. If you keep hammering on these receptors, say with too much nicotine, they'll desensitize. They basically just give up and shut down for a while.
Okay, on to the muscarinic family. And I've got a killer mnemonic to help you remember the G proteins, because this is always on exams. Just remember the pattern QIQ. The odd-numbered receptors, M1, M3, and M5, they're all coupled to GQ. That's the Q. GQ fires up the PLC pathway, which cranks up intracellular calcium. The even-numbered receptors, M2 and M4, are coupled to GI. That's the I. GI inhibits adenylate cyclase, which drops the levels of CMP. So just think odds are Q, evens are I. QIQ. It's a lifesaver for keeping these pathways straight.
Okay, time to zoom out. We've been looking at the tiny details in the synapse. Now, in Section 4, we're going to look at the big picture, the major cholinergic pathways. Where do these neurons actually live in the brain and the body, and what huge systems do they control? In the central nervous system, there are two big cholinergic highways you need to know about. First up is the basal forebrain, and specifically a little spot called the nucleus basalis of Maynard. These neurons spray acetylcholine all over the cortex, and they are absolutely essential for things like attention, learning, and forming memories. In fact, this is the exact group of neurons that dies off in Alzheimer's disease, which is why memory is so affected. The second major system is in the brainstem. These guys project up to the thalamus and basically act as the switch that controls our sleep-wake cycles and keeps us alert.
Now, outside the brain, in the periphery, acetylcholine is just everywhere. It's a major player. In the somatic system, the one you control consciously, it's the final command at the neuromuscular junction that makes your muscles move. But its role in the autonomic nervous system is even bigger. And if you remember one thing from this section, make it this. ACH is the neurotransmitter for all preganglionic neurons. Doesn't matter if they're sympathetic or parasympathetic. And it's also the transmitter for all postganglionic parasympathetic neurons. It's truly a master controller out in the body.
All right, let's bring it home. This final section is pure 24-karat high-yield gold for your exams. We are going to tackle one of the most classic, tricky questions they love to ask, and then I'm going to give you a rapid-fire summary of all the organ effects you need to have memorized. So grab your coffee, focus up. This is the good stuff. I would bet money you're going to see this question in some form. It's a classic pharmacology paradox. We're taught that the parasympathetic system, rest and digest, lowers blood pressure. But here's the kicker. The parasympathetic nerves don't even touch the smooth muscle in most blood vessels. So what gives? How can acetylcholine cause vasodilation if there's no wire connected? And the answer is just, it's beautiful because it's an indirect effect. Check this out. The acetylcholine floating in the blood doesn't talk to the muscle cell. No, it talks to the super thin layer of endothelial cells that line the inside of the blood vessel. It binds to their M3 receptors. This kicks off that GQ pathway we talked about, which jacks up the calcium inside the endothelial cell. That calcium then turns on an enzyme called eNOS, which stands for endothelial nitric oxide synthase. eNOS then produces nitric oxide, which is a gas, and that little gas molecule just diffuses right next door to the smooth muscle cell and tells it to relax. It's this elegant, indirect bank shot. So cool. Okay, and here it is. This is your screenshot this right now slide. This is your last-minute cram sheet for the organ effects. Let's do a quick lightning round. Muscarinic activation. What does it do? In the eye, meiosis, pupil constriction. In your glands, more secretions. Think drooling, sweating, crying. Bronchi, constriction and more mucus. The heart, that's the M2 receptor. It slows the heart rate down, bradycardia. GI tract, more motility, more secretions, gets everything moving. Bladder, contracts the main muscle, relaxes the sphincter, it makes you pee. And the blood vessels, remember, that's the indirect one, vasodilation through nitric oxide. Basically, if it sounds like rest and digest, that's acetylcholine's job. If you can nail this list, you're going to feel so much more confident on your pharm exam.